Hey Mustangs, in this video we're going to be taking a look at dihybrid crosses. Now if you understood monohybrid crosses, which is what you see here, where you're just looking at one trait and you're taking parents and you're doing a cross between those two parents for just one single trait, you're going to be able to get dihybrid crosses pretty easy. It'll be a piece of cake. Okay. Now in order to really understand dihybrid crosses, you have to understand two laws. You have to understand the law of segregation and the law of independent assortment. So let's go ahead and take a look. Now, we went over the law of segregation when we did monohybrid crosses, and you can actually see it happening here. Okay? If you take a look at this here, this parent, the male plant, was heterozygous, and we put one of its alleles here and the other of its alleles here. And the reason why we separated it over two boxes was because of the law of segregation. Same thing with the female. We separate it between the two boxes because we're showing the law of segregation, saying that they're going to wind up in different gametes. In the female's case, the alleles will separate. This will wind up in one egg. This will wind up in another. In the male, this will wind up in one sperm. This will wind up in the other. So in order to understand dihybrid crosses, let's go ahead and take a look at the first example. Make sure that you have your notes from class. The two examples in the notes will be used to help you understand dihybrid crosses. So we take a look here at example 1. In pea plants, yellow seeds are dominant to green seeds. Round seeds are dominant to wrinkled seeds. Cross a male who is heterozygous for both traits with a female who is heterozygous for both traits. Now when we look at this, in a dihybrid cross, di means two. So instead of following just one trait, like the color of the seeds, we're going to follow two traits, the color of the seeds and if they're round or wrinkled, so the texture on the outside of the seeds. So looking at this, first thing we want to take into consideration is what letters we're going to be using. So for yellow seeds, we'll use a capital Y. For green, since yellow is dominant over green, we'll use a lowercase y. Make sure that you make a big distinction between the dominant versus recessive in the case of y because the capital looks very similar to the lowercase. In trait 2, the second trait we're looking at in this dihybrid cross is round seed, and round is dominant. So round allele is dominant over the wrinkled allele, so we use capital R and then lowercase r for the wrinkled. Now we got to put together the parent genotypes. So up here we say cross a male who is heterozygous for both traits with a female who is heterozygous for both traits. Okay, so the male, he's heterozygous for seed color, so he's big Y, little y. And he's also heterozygous for the seed texture, so he's big R, little r. Here for the female, she's heterozygous for both as well, so she's big Y, little y, and big R, little r. Okay, so we follow the exact same principles that we did in a monohybrid cross. Now, looking at the law of segregation and the law of independent assortment, what we're saying here when we look at the parents' genotypes, big Y, little y, big R, little r. Now both parents are that, so this slide here will cover both parents and what's going on with the law of segregation and law of independent assortment. So looking at this here, we're going to draw the chromosomes. Now the letters that we see in a genotype represent alleles that are found in the genes inside the chromosomes. So here, let's say that right here is the gene for seed color. So on this parent we see they have a dominant and a recessive. So that means from their parents they received a dominant allele from one parent and a recessive allele from the other. So that's within the DNA inside the chromosomes there. So this has instructions for making yellow seed color, and this has instructions for making green seed color. And let's say on another pair of chromosomes we have the gene for the seed texture, and we know based on the genotype that this plant got a round allele from one parent and a wrinkled allele from the other. So that's really what's going on when we write the parents' genotypes like this. And we know that one of these chromosomes, this is of course a homologous pair, one of these came from mom, one of these came from their dad. Same thing here with these chromosomes. One of these came from their mom, one of these came from their dad. Now, according to the law of segregation, we know that they cannot wind up in the same gametes at the end. So if this cell were to go through meiosis, the law of segregation says that homologous pairs must separate into different gametes. So here, what would happen? These would have to separate. So it would look something like this. Let's say this one here winds up in this, let's say egg in this case. So there's the big Y. And then the other one from this homologous pair winds up over here. And this is a little y. Okay, oh, almost looks the same. So this is a big Y, I'll make it bigger. Big Y, and there's a little y. Okay, so this one wound up here, this one wound up there. That follows the law of segregation. So we know that if this plant were to make its gametes, eggs in this case, that the law of segregation would cause these two chromosomes to separate. Same thing is true for this homologous pair. These have to follow the law of segregation. They have to wind up in different eggs. Now, the question is this one here going to wind up over here? So the two dominant, are they going to wind up together? Is this one going to wind up over here with the two recessive together? So would it look something like this? Big R, and this one with the little r, so the recessive move together? Or does the little r wind up over here and the big R wind up over there? Okay. Now, according to the law of independent assortment, it can be either one. Okay, so it's possible that this one winds up over here, or it's possible that this one winds up over here. So they can actually be switched. So there's actually different possibilities, different combinations. It could be big Y, little r, or big Y, big R. Over here it could be little y, big R, little y, little r. So the law of independent assortment says that the way these chromosomes separate following the law of segregation is completely random. And this is important to understand because there's different possible combinations. Like I said, it could be big Y, big R, big Y, little r could have wound up in here. Okay, so keeping this in mind, let's go ahead and take a look at our Punnett square. So in a dihybrid cross, instead of just 4 boxes, we have 16 boxes that we are going to deal with. So in order to follow the law of segregation and the law of independent assortment, we follow this rule here. So I'm going to use red here. We're going to say that one possible combination is big Y can wind up with big R. So it would look like this. We put that possible combination here, big Y, big R. Another possible combination is that big Y can wind up in the same gamete with little r. So we'll put that right here, big Y, little r. Well, there's another possible combination. Little y could wind up with big R. So little y, oops, little y, big R. Or little y can wind up with little r. Little y, little r. So that's one possible combination here for this parent. The other parent, same genotype, so it's going to be the same progress here, or same process. So here, big Y can wind up with big R. Big Y can wind up with little r. Then little y with big R. And finally, little y, little r. So those are all the possible gamete combinations. Okay. So remember, this is basically like these are the separate sperm that would have been created here with all the possible combinations based on the law of segregation and law of independent assortment. So this would be one sperm, this would be another possible sperm and another. Over here, oh, I forgot to write this. So we're saying that this is the male up here on this side and all the possible sperm that he could create. And over here would be the female and all the possible eggs that she could create. So we use the same principles that we had with the monohybrid crosses. So here in this first box, if the sperm carrying these alleles got together with the eggs carrying these alleles, you'd wind up with big Y, big Y, big R, big R. And if we use the principle of dominance here, Y represents yellow, capital Y represents yellow, and capital R represents round. So this offspring, from this sperm and this egg coming together, this offspring would wind up yellow and round. Okay, we go to the next one. Big Y, big Y, big R, little r. So again, yellow and round, the offspring. Here we have big Y, big R, big R. And we just keep on going, so I'm going to pick up the pace a little bit, go a little bit faster here. And here we have our first case of yellow and wrinkled instead. If you want, you can fill yours in, pause the video, fill it in, and then fast forward past the rest of me filling this in. All right. So now that we have this all filled in, we can actually go through and use the principle of dominance to determine whether the offspring that represents the gametes coming together in each box, whether they're going to be yellow, green, or if they're going to be round or wrinkled. Now, for the ratios. Okay. In a monohybrid cross, there was always two possible phenotypes when we looked at the monohybrid crosses that we looked at. Here in a dihybrid cross, there's actually 4 possible phenotypes. So the possible phenotypes are: the offspring could be yellow with round seed shape, so that's one possibility. It could be yellow with wrinkled. Could be green with round, or it could be green with wrinkled. Okay. So if we take a look at what we got in our Punnett square and we look for these different phenotypes, we find that there are 9 offspring that come out with yellow and round. There are 1, 2, 3 that come out yellow and wrinkled. 3 that come out green and round, and 1 that comes out yellow and wrinkled. So that would be our ratio, and we could write it as a 9 to 3 to 3 to 1. So that's our expected phenotype ratio if we were to cross 2 heterozygotes in a dihybrid cross. Now, go ahead and try example number 2, and we'll be checking that in class.